this lecture, we are going to talk about part 4 of translation, and we focus on termination. Main points of the lesson are stop codons and release factors. In the previous video, we discussed about elongation that includes the matching tRNA anticodon with messenger RNA codon, peptide bond formation and translocation of ribosomes. In this lecture, we focus on termination. Termination is the final stage of translation. Termination of translation occurs when nonsense codon, that is UAA, UAG or UGA is encountered. At the time of aligning with A site, these nonsense codons are recognized by release factors in prokaryotes and eukaryotes. So, in termination, synthesis of polypeptide chain is terminated by release factor. What is a release factor? It is a protein that recognizes the stop codon at the end of coding sequence. When the release factor binds to the A site, the bond between the tRNA and last amino acid of polypeptide chain, this bond breaks in the P site, and the release factor hydrolyzes the bond between the polypeptide chain and the tRNA, causes the release of polypeptide chain from the tRNA molecule in the P site. The remaining parts of translation complex dissociate. Each mRNA molecule is translated only a limited number of times before it is destroyed. Let me briefly restate the main points. Termination is the final stage of translation, and termination of translation occurs when a nonsense codon, that is UAA, UAG or UGA, that is encountered in the A site. At the time of aligning in the A site, these nonsense codons are recognized by the release factors in prokaryotes and eukaryotes. So, the synthesis of polypeptide chain is terminated by the release factor.